Welcome back friends, welcome back in another video tutorial. So today we will going to talk about mixed acid fermentation. So mixed acid fermentation is a type of fermentation which is a property of Enterobacteriaceae. So here mixed acid, the name mixed acid suggested that after this fermentation end product will be different type of acids, right? So these acids are lactic acid, acetic acid, formic acid, succinate and ethanol. Okay, and also there may be some cases where gas will be produced. Okay, remember it very carefully that all bacteria that can do mixed acid fermentation does not mean that they will going to produce gas. Only those mixed acid fermenters will going to produce gas that have formate dehydrogenase. These formate dehydrogenase have the ability to break formate into gases. Okay, so that is the idea of mixed acid fermentation. So there is another thing which is associated with mixed acid fermentation is butanidiol fermentation. Butanidiol fermentation is done by non-fecal enteric bacteria. Okay, for example, Klebsiella and Enterobacter. They will form all the mixed acid fermentation end products. Along with that, they may also produce gases if they had formate dehydrogenase, and along with that, as a addition, they have the ability to produce 2,3 butanidiol. Okay, from the condensation of 2-pyruvate. Now remember it very carefully that this butani in this butanidine fermentation there is very low amount of acid production. Okay, not only that, in this butanidine formation, not formation, sorry, in this butanidine fermentation, there will be formation of a distinctive intermediate known as acetoin. So, the low acid amount as well as the presence of acetoin will help us to distinguish all the butanidyl fermenter from the other mixed acid fermenter, and this test will going to help us to distinguish between the non-fecal enteric bacteria and the fecal enteric bacteria. Okay, so this is very important, and we will going to talk about this elaborately during our medical microbiology lecture series, which we will going to make in some other times in the future. So that is the basic idea of mixed acid fermentation. So mixed acid fermentation is usually for Enterobacteriaceae organisms, right? So here the end products are mixture of acid, for example, lactic acid, acetic acid, ethanol, formate, succinate. Okay, and if this the bacteria who are mixed acid fermenters have formate dehydrogenase, then they can also break the formate to produce gases. Okay, so remember it very carefully. I am saying this to you repeatedly that all mixed acid fermenters are not gas producers, right? But all gas producers are mixed acid fermenters. Okay, so along with that, we have also talked about another type of fermentation, that is butanidyl fermentation, which is done by especially non-fecal enteric bacteria, for example, Klebsiella and Enterobacters. So in this type of fermentation, what will happen? In this type of fermentation, all the end products of mixed acid fermentation will occur. Even gas production also occur if they have formate dehydrogenase, and along with that, there will be formation of 2,3 butanidiol. And later I have told you that this butanidine fermentation there is a very low amount of acid production, and also there will be a distinctive intermediate named acetoin. So these two things, low amount of acid production and acetoin, help us to distinguish these butanidine fermenters, which are mainly non-fecal enteric bacteria, from fecal enterics, which are mainly mixed acid fermenters, right? So I hope this video will be helpful for you. Thank you.